Welcome to module number 11, where I will talk about the origins and evolution of early Homo. We are on our way of learning about the lifeways of our ancient ancestors, so let's get started. In 1891, no fossils of Australopithecus had yet been found. In fact, the only fossil humans that had been discovered were Neanderthals, dismissed by many simply as pathological modern humans. Just 20 years earlier, Charles Darwin had published The Descent of Man, in which he formulated many of his ideas about human evolution. If Darwin was right, then scientists should eventually discover fossils of creatures that are not quite human and not quite ape. Though Darwin hypothesized that these fossils would be found in Africa, other scholars, such as Ernest Haeckel, argued that they would reside in Asia, given the close similarities between humans and orangutans. Remember, DNA had also not been discovered yet, so there is no way of knowing which of the apes was most closely related to humans. Haeckel not only hypothesized the existence of this half-man, half-ape, he even gave it a name, Pithecanthropus erectus. The writings of Darwin and Haeckel attracted the attention of a young Dutch anatomist named Eugene Dubois, pictured at the upper left. Dubois became obsessed with finding fossils of these early humans, and in 1887 he moved to Indonesia, which was at the time a Dutch colony. You can see the map at the bottom left. 4 years into his search on the banks of the Solo River near a village called Trinil, Dubois found a tooth, a partial skull, and a femur. The femur indicated that this creature walked on 2 legs, but the brain was only about 1,000 cc's. It was too small to be a human brain and too large to be from an ape. It was exactly what he was hoping to find, and Dubois named it Pithecanthropus erectus. We know these fossils by a different name, Homo erectus, though, as we have already learned, the earliest hominins are from Africa, not Asia, and these fossils were an important first step in our understanding of the evolution of our own genus, Homo. In this module, I will discuss the evolutionary changes that characterize the earliest members of our own genus. How do we know we've discovered a fossil from Homo and not from Australopithecus? Next, we will look at fossils from the very earliest members of our own genus. These are typically categorized into the species Homo habilis, though there is some evidence that there may have been 2 different species of early Homo coexisting in Africa. Soon after the evolution of our genus, a different kind of hominin evolved, Homo erectus. Homo erectus is what Dubois found on Java, and we will look at this fascinating global species in detail. In particular, we will look at the evolutionary trends that happen in the Homo erectus lineage and how anatomical and behavioral adaptations in this species set the stage for evolution, not only of our genus Homo, but of our species Homo sapiens. Let's dive in with a look at the first species of Homo, Homo habilis. Humans have exceptionally large brains, and we rely on material culture to survive. These 2 characteristics have turned out to be quite important in defining the genus Homo. Although we find stone tool technology in the smaller-brained Australopithecus genus, it is a combination of stone tool culture, larger brain size, and smaller tooth anatomy that can be used to identify fossils from our own genus. Of course, the evolution of our genus did not happen overnight, and so there are transitional fossils older than 2 million years that bridge the anatomical gap between Australopithecus and Homo. Some researchers call these fossils Homos, and some call them Australopithecus. Of course, that is exactly what you would expect for an evolving lineage. The geologically oldest fossils that fit the criteria for inclusion in the genus Homo were initially discovered by Louis Leakey in Olduvai Gorge, Tanzania in the 1960s. The brain was larger than that of an Australopithecus, around 600 cc's. The teeth were smaller and the landscape was littered with Oldowan stone tools. In fact, these tools were evidence of a dietary change that would have selected for smaller rather than larger teeth. This dietary shift may also have been critical in providing the energy necessary to grow a large brain. In other words, these features are probably linked to one another. Leakey and his colleagues named this new creature Homo habilis, which means handyman. Two of the fossils found by Leakey are shown above and to the left, the skull OH24 and the mandible OH7. To the right is another Homo habilis skull discovered in Kenya. Additional finds of Homo habilis have revealed that this species probably had an Australopithecus-like body with relatively long arms and short legs. However, there is some intriguing evidence that Homo habilis was not the only species of early Homo existing at this time. Shown here is a skull discovered in Kenya in the 1970s that has recently been redated to a bit over 2 million years. It has a larger cranial capacity than most specimens attributed to Homo habilis, and the shape of the face and the tooth rows are a bit different from what is found in most Homo habilis fossils. There are many researchers who regard this as a different species and call it Homo rudolfensis. A recent discovery of additional fossils in Kenya support this idea that two different kinds of early Homo coexisted. Others regard these differences as normal variations one would find in a population of hominins. Figuring out which of these hypotheses is correct is not as easy as it sounds. There is tremendous variation within humans today, and that variation is not because there are different species of humans. At the same time, there are different species of mammals, like squirrels or antelopes or macaque monkeys, whose skeletons are not substantially different from one another. Are we perhaps underestimating the number of species in the fossil record? For the time being, let's proceed as though these are all one species, Homo habilis, but keep in mind that the situation might be a tad more complex than that. Homo habilis fossils reflect a major change in how hominins utilize their landscape. Oldowan stone tools are quite common at Homo habilis fossil sites. The anatomy of the skull of Homo habilis shows a significant reduction in the size of the chewing muscles, face, and teeth, in part because the stone tools were doing much of the work the teeth and jaws had previously done. Additionally, Homo habilis hand bones appear well adapted for stone tool construction and use. They had larger brains, but their bodies remained small, with relatively long Australopithecus-like arms. Driving many of these anatomical changes was an adaptive shift to surviving on a landscape that was becoming more and more grassland. The use of tools and the environments they inhabited may indicate that the earliest member of our species were adopting a more varied, generalized diet of both plants and meat. This dietary flexibility would have helped them survive in difficult times, and it would have helped fuel a growing brain. Around 1.8 million years ago, early Homo evolved into a new kind of hominin, with an even larger brain, even smaller back teeth, and long, human-like legs. This is Homo erectus. Homo erectus fossils are characterized by other features like large brow ridges and long, low skulls. But unlike every hominin discovery up to this chronological point in human evolution, fossils of this species are not found exclusively in Africa. Homo erectus expanded both inside and outside Africa and quickly inhabited Europe and Asia, as illustrated on the map to the right. Homo erectus is therefore the first global human species. We, of course, already encountered Homo erectus outside Africa in the Trinil skull, shown on the upper left, discovered by Eugene Dubois in 1891. But the current holder of the title of the oldest hominin outside Africa belongs to the skull at the bottom left from the site of Dmanisi, a 1.77 million year old fossil site in the former Soviet Republic of Georgia. Nevertheless, let's start in Africa and look at the evolution of Homo erectus on that continent. The image here shows just how different Homo erectus is from an Australopithecus. Keep in mind that this robust Australopithecine lived at the very same time as Homo erectus. These two species, both upright walking hominins, would have met one another on the grasslands of East Africa, but they were quite different from each other. Australopithecus possessed large chewing muscles anchored to the sagittal crest atop the skull. The premolars and molars were enormous, helping the species eat a diet of hard nuts, raw tubers, grasses, and seeds. In contrast, the skull of Homo erectus, shown at the bottom of the slide, lacks a sagittal crest, has a much larger brain, and much smaller teeth. These adaptations reflect a more diverse, softer diet that included meat. Homo erectus also had a larger body size and longer legs than Australopithecus and Homo habilis. Much of what we know about this species derives from a remarkably preserved skeleton of a juvenile male discovered in the 1980s at a site in Kenya called Nyarikitome. On the left is the Nyarikitome Homo erectus skeleton. It is 1.6 million years old and remarkably complete. Unlike Australopithecus and Homo habilis, this skeleton has human-like limb proportions. In other words, the legs are quite long and the arms are quite short. This anatomical change indicates that Homo erectus walked just like modern humans do, and that they were no longer climbing trees. The skeleton is from a young boy, perhaps only 11 years old or so at the time of his death, but he was already 5 feet 6 inches tall and probably would have grown more, perhaps reaching 6 feet. Thus, unlike previous hominins, Homo erectus was quite large. Additional fossil discoveries have revealed diversity in body size, however, so not all Homo erectus individuals were this big. The skull of the Nyarikitome boy is shown on the right. He has a relatively large brain, about 900 cc's. At this size, his brain was larger than the typical Homo habilis, roughly 600 cc's, but smaller than a modern human, which is 1400 cc's. In fact, the cranial capacity of this Nyarikitome boy is exactly halfway between those of the average chimpanzees and the average human. One question that the Nyarikitome juvenile skeleton allows us to address is whether Homo erectus grew slowly, as modern humans do, grew quickly, as modern apes do, or grew at an intermediate pace. As I discussed in a previous lecture, humans are unique in having a very slow growth period from birth to adulthood. Additionally, humans are unique in having an adolescent growth spurt. Did Nyarikitome have a growth spurt? To know this, scientists have to first determine how old he is when he died. Because teeth grow and erupt in predictable patterns in humans, scientists can use the development of the teeth of the Nyarikitome individual to infer his age. Notice that the tips of the roots are not yet completely developed in some teeth. By human standards, he would have been about 11 years old at the time of his death, but that assumes he was growing slowly like a human. To test this, scientists counted the growth increments of the developing teeth, called pericomata, shown in the image to the right. Because pericomata are laid down every 8 to 9 days, by counting the lines, scientists can determine how much time it took to grow the tooth, and in turn determine the age of Nyarikitome. Using this method, his age at death was determined to be 8 to 9 years old. This finding suggests that Homo erectus was developing faster than a modern human, though still slower than a modern ape. In other words, even the growth of Homo erectus was transitional between ape and human. Scientists of human evolution often think of an increasing brain size as a positive selective force, with no negative consequences. However, with all evolutionary changes, there are trade-offs, and an increase in brain size is no different. Not only are large brains energetically expensive, but they must be birthed through female pelvises that have evolved a smaller relative size to accommodate the biomechanics of upright walking. And not only is there a tight fit between the size of the newborn's head and the width of its shoulder and the pelvic outlet, but the shape of the opening changes from the pelvic inlet to the midplane and outlet, causing the baby to corkscrew through the birth canal during delivery and ultimately face backwards. This rotational birth is unique to humans and may necessitate the presence of midwives and other helpers during human birth. So what was childbirth like for early hominins and for Homo erectus? The shape of the female pelvis and the estimated size of the newborn hominin provide some insights. Childbirth in Australopithecus is inferred from the pelvis illusi, shown on the right. Anthropologists have suggested that while Australopithecus may have had discomfort during birth, rotational birth may not have been present yet. Similarly, a new fossil pelvis of Homo erectus indicates that birth was probably difficult, but not quite human-like, even in this species. Let's return to the fossils of Homo erectus and investigate some interesting new findings. Not only can we use the skeletal anatomy of Homo erectus fossils to infer how they walk, this species left us fossilized footprints. At the 1.5 million year old site in Kenya called Ilere, scientists have recently unearthed a large collection of footprints made by Homo erectus. These prints shown to the right are nearly indistinguishable from prints from a modern human would make, indicating that this species moved essentially the way we do. The feet are arched and the big toe is in line with the other toes, as is found in human feet. So although the brain was still outside the range found in modern humans, the mode of locomotion was essentially identical. There's also a skull from Ilere, shown from top view, bottom left, that is smaller and has smaller brow ridges than many other fossils attributed to Homo erectus. This is skull KNM-ER 42700. This means that this is the 42,700th fossil discovered at this locality in Kenya. Researchers have recently exceeded 60,000 fossils. Of course, not all of these are hominins, but it indicates just how numerous fossils can be in some of these localities. The anatomy of the Ilere skull indicates that Homo erectus was quite variable. It may have maintained a high Australopithecus-like level of sexual dimorphism. Shown here are three more skulls of African Homo erectus. The top image to the left is a fossil from 1.2 million year old deposits at Olduvai Gorge, called OH9. Notice the enormous brow ridge and a large area for muscle attachments on the back of the skull. The skull of the specimen also has very thick bone. Below OH9 is the Daka skull, a 1 million year old fossil discovered by Tim White's team in Ethiopia. The importance of the skull is that it looks in many ways like Homo erectus fossils discovered in Asia, which we'll learn more about in just a moment. There were some researchers who were beginning to think that the African Homo erectus fossils were different enough from the Asian Homo erectus fossils to call them a different species. They even had a name for this species, Homo ergaster, by finding a specimen, Daka, that bridges the gap between these fossils. There is more support for the idea that Homo erectus is just one variable species. To the upper right is a later Homo erectus fossil, also from Ethiopia, called Bodo. It is 600,000 years old and has a brain size that is within the range of what is found in humans today. It is 1 million years younger than some of the first Homo erectus fossils we started with, like the Nerekotomb juvenile from Kenya. And Bodo demonstrates that brains were getting bigger this whole time. One neat thing about this skull was the discovery of deliberate cut marks on it, shown to the bottom right, that indicates that Bodo was defleshed after his death, either ritually or due to cannibalistic activities by Homo erectus. From earlier lectures, remember that evolution in general, and human evolution in particular, can only be fully understood in the context and the environment these ancient creatures inhabited. There were many changes from Homo habilis to Homo erectus, and many changes within the evolving lineage of Homo erectus. One way to understand why these changes occurred is to accurately reconstruct the ecology of these hominins, like Homo erectus. Shown here is an artistic reconstruction of Daka, Ethiopia, approximately 1 million years ago. The fossils collected at this locality indicated that Daka was a sprawling savanna grassland environment, with patchy wooded areas. The inhabitants included large predatory cats, monkeys, the ancestors of modern zebras, giraffes, elephants, hippos, rhinos, warthogs, and African rodents. Note the large number of antelope species, and of course, in the distance, our own ancestors, Homo erectus. As we have already discussed with regard to Eugene Dubois' 1891 discovery of the Trinil Homo erectus skull on the Indonesian island of Java, this species expanded its territory beyond the borders of the African continent. Interestingly, almost immediately after Homo erectus evolved, presumably in Africa, it spread east into Asia, which you can see on the map at the bottom right. Now, 500,000 years later, Homo erectus expanded west into Europe. This expansion indicates that Homo erectus was adaptable and had evolved the intelligence to use resources in its environment to make the tools necessary to survive. Some of the oldest fossils of early Homo erectus found outside Africa are from the 1.77 million year old site of Dmanisi, which is in the former Soviet Republic of Georgia. Dmanisi has turned out to be one of the most productive early Homo fossil sites ever found, with several skeletons and 5 skulls, including the one shown here. The skulls have cranial capacity in the 600 to 750 cc range, which incorporates the range of both Homo habilis and Homo erectus. Like Homo erectus, the Dmanisi individuals also had long legs compared to their arms. Since Dubois' work in the late 1800s, many other paleontologists have worked on the island of Java and found many more fossils of Homo erectus, including over a dozen partial skulls. The most complete fossil skull is shown here, Sangiran 17. Notice the long, low skull, with a thick brow ridge and flat surface for neck muscles in the back. Also, notice the relatively large face with enormous cheekbones. Sangiran 17 has a large brain of 1000 cc's, which is not quite within the range of most humans today, but certainly larger than any ape or Australopithecus. There is also a thick sagittal keel across the top of the skull. A sagittal keel is a thickening of the cranium down the midline of the skull. Do not mistake this for the sagittal crest. It is very different since the temporalis muscle does not anchor to a sagittal keel, as it does a sagittal crest. Instead, the sagittal keel may just be a thickening of bone, helping to resist the bending forces of the skull during chewing with the front teeth. In many ways, this Indonesian skull looks like the skulls of similarly aged Homo erectus back in Africa, like OH9 and Dhaka, indicating that there was gene flow connecting these populations and maintaining a single Homo erectus species. There are also wonderful fossils of Homo erectus from several sites in China. The 3 important sites are shown on this map. There are not any hominins yet from Majiao, although stone tools and amidal bones with cut marks on them indicate that hominins were active in this area 1.7 million years ago. Gongwengling preserves a 1.2 million year old Homo erectus skull with a brain size of 800 cc's. But the most productive Homo erectus site is Zhoukoudian, a cave not far from Beijing, preserving what many call the fossils of Peking man. To the far left is Peking man, the common name given to fossils of Homo erectus from the Zhoukoudian locality in China. In the 1920s and 30s, remains of up to 40 individuals were discovered, including several nearly complete skulls. An image from this excavation is shown at the bottom left. These fossils date to over 500,000 years, with very recent dating putting them at about 750,000 years. Unlike other cave localities we have discussed in this class, in which the fossil bones of hominins were washed into caves or inadvertently dumped into caves by carnivores, the Zhoukoudian caves, shown forming on the right, were places that Homo erectus actually lived. We know this because these caves preserve the remains of stone tools and evidence of controlled fire. Fortunately, the great paleoanthropologist Franz Weidenreich made detailed copies in the form of cast of these Peking man fossils that he distributed all over the world, and he published detailed anatomical descriptions of these fossils. This is fortunate because during World War II, the entire fossil collection was lost. Many recent efforts have been made to rediscover where these fossils may have ended up, but none have been successful. The fossil record seems to indicate that early Homo erectus populations migrating out of Africa generally took a right turn and headed east into Asia. However, there is now evidence from cave sites in Spain that by at least 1.2 million years ago, these hominins started to turn west into Europe. One of the oldest sites that has yielded hominin fossils in Europe is called Grand Dolina, which is about 900,000 years old. Shown here are excavations happening at Grand Dolina, shown on the upper images, and a map of where this locality is, shown on the lower left. The image at the upper right is the face of one of the many hominins found at Grand Dolina. This is a face of a child, though plenty of adult bones were recovered from Grand Dolina as well. In many ways, these fossils are like those of Homo erectus found elsewhere. Differences, however, the nose is wider and there is increased robusticity. Other fossils from Italy, Germany, and England are also Homo erectus-like in many ways, but are different in others. Interestingly, these European fossils began to resemble fossils found in younger sediments in Europe. It appears as though Homo erectus migrated into Europe and became somewhat isolated from the rest of the Homo erectus population. This isolation led to anatomical differences in these hominins and eventually led to the group we now call Neanderthals. But does that mean they evolved into different species? Some think so and regard these fossils from Grand Dolina as a new kind of hominin, Homo antecessor. Others think probably not and regard these as a regional variant of Homo erectus. Let's review. By the time we reach Homo erectus, especially later Homo erectus living about 500,000 years ago in Asia, Africa, and Europe, these hominins began to closely resemble you and me. They have brains that are within or very close to the low range of human brain size today. They have small teeth and rely very heavily on material culture to survive. They have controlled fire and cook their food, something I'll discuss in just a moment. And they have long legs with a striding bipedal gait indistinguishable from yours and mine. Many of the biological features we find in humans today evolved not with our own species Homo sapiens, but with the immediate ancestor of Homo sapiens, Homo erectus. As is illustrated here, compared to Homo habilis, Homo erectus evolved smaller teeth and smaller jaws. The brain increased in size, although it was housed within a very robust skull with thick bones and a prominent brow ridge. And Homo erectus evolved a larger, human-like body size with an increase in leg length. We spent this entire lecture characterizing these changes, but we have not yet addressed one key question. Why did this happen? Most likely, what drove all of these changes were improvements in diet and in how these hominins accessed resources from their environment. Homo erectus was a hunting hominin and ate more meat than earlier hominins. Though meat eating started long before Homo erectus, these earlier hominins were mostly scavenging, since the stone tool cut marks are often found on top of bite marks made by carnivores. See the images at the top. Meaning that carnivores made the kills and hominins scavenged what they could. Homo erectus is often associated with different, more sophisticated tools than the Oldowan tools found with later Australopithecus and with Homo habilis. These stone tools are called Acheulean. The hand axe is the most common Acheulean tool type. It is shown in the bottom right image next to a site in Kenya called Ologolesti, where hand axes litter the landscape by the hunters. The artistic reconstruction of Homo erectus butchering a carcass is certainly imagined, but it is informed by fossil evidence. The hand axe can be used to cut and scrape the meat away from an animal carcass, like the zebra shown here. We know of this because these tools are often found in association with the fossil skeletons of very large, butchered animals, like hippopotamus, zebras, and elephants. Now, keep in mind that Homo erectus was not only a carnivore, but also relied heavily on gathering tubers, nuts, berries, and many other foods to survive. Most likely, Homo erectus lived in groups and shared with one another the foods they gathered and hunted. This increase in food quality and available energy fueled the growth of an energetically expensive brain. Even though brains are only about 2% of the body mass, they eat up 20% of the body's energy. Having a large brain, if nutrients are scarce, could be detrimental to an individual and could lead to selection against large brains. Large brains can only evolve if there is ample nutrition to support such an energetically expensive tissue. This happened with Homo erectus, and it created a feedback loop whereby the larger-brained individuals were more intelligent and could make more effective material culture to access resources from their environments and support the enlarged brain. This is one of the best examples of how culture and biology interact in human evolution. One final and critically important cultural innovation of Homo erectus was controlled fire. Fire is critically important because it allowed Homo erectus to stay warm and to inhabit cooler climates that otherwise would have been inhospitable. Additionally, it allowed these hominins to cook their food. Cooking softens food and makes it more digestible than raw food. Cooking was thus a critical human adaptation that allows us to access more nutrients and calories from our food in a more rapid and physically easier manner. There is now evidence from the South African cane of Wonderwerk, shown on the right, that Homo erectus had controlled fire by 1 million years ago. Let's wrap up this lecture with a final review of Homo erectus. We begin to find fossils of this species at sites dated around 1.8 million years ago in Africa and Asia. Homo erectus spread throughout the Old World and populated remarkably diverse areas, ranging from South Africa to Spain to Indonesia and everywhere in between. Their success was largely dependent on their ability to use material culture to survive in these diverse ecosystems. They used more sophisticated tools called Acheulean, and by 1 million years ago had controlled fire for cooking and warmth. These cultural innovations affected the biology of Homo erectus, leading to the evolution of larger brains, smaller teeth, and smaller jaws and chewing muscles. They evolved larger body size and limb portions that were identical to humans today. One final thing to note is that the earliest Homo erectus fossils from Africa and Dmanisi looked very different from the later Homo erectus fossils from Bodo and from Zhuge-Dan. Some researchers feel these differences warrant the naming of different species, but other researchers argue that all we are seeing is evolution within a species over time. Gradually, brains get bigger, teeth get smaller, skulls got thicker, and later got thinner. These changes mirror the technological advances made by Homo erectus and illustrate the intertwined relationship of culture and biology in these hominins. Many of these changes we see in Homo erectus foreshadow the evolution of our own species, Homo sapiens, which will be the subject of my next lecture.